Hello and welcome to the Midnight Lab. Before we begin tonight's exploration, take a moment to like the video and subscribe to the channel if you enjoy calm, thought-provoking journeys through science and the mysteries of the universe. And while you're here, let me know in the comments where you're watching or listening from tonight. Whether you're in a quiet apartment, a late-night office, a train moving through the dark, or lying in bed somewhere across the world. It's interesting to remember that each place exists only because your brain is constructing it for you right now. The room around you, the sound of this voice, even the sense that you are here, listening, all of it is being created inside your mind. And that raises one of the strangest and most fascinating questions in all of science. How does the brain create a mind? Inside your skull sits an organ weighing a little over 1 kilogram. It's soft, slightly wrinkled, and made of ordinary biological material, cells, chemicals, and electrical signals. There is nothing visibly magical about it, and yet somehow this biological machine generates thoughts, emotions, memories, dreams, and the feeling of being a conscious self experiencing the world. Every sight you see, every sound you hear, every idea you've ever had, they all exist because of patterns of activity inside this organ. At first glance, that idea almost sounds impossible. How could billions of tiny cells, communicating with microscopic pulses of electricity and chemistry, produce something as rich and mysterious as the human mind? And yet, that is exactly what modern neuroscience suggests. Your brain contains around 86 billion neurons, each one connected to thousands of others, forming an immense living network. Within this network, signals flash across trillions of connections every second. From this vast electrical conversation emerges perception, memory, imagination, and somehow awareness itself. But here's where the story becomes even stranger. Your brain doesn't simply record reality like a camera. It doesn't passively observe the world. Instead, it actively constructs an internal model of reality, constantly predicting, interpreting, and filling in the gaps of the information it receives. In other words, the world you experience is not the raw universe outside your skull. It is the brain's best interpretation of it. The colours you see, the feeling of time passing, the sense that you exist as a single continuous self. All of these may be carefully constructed illusions produced by the brain's incredible machinery. And despite centuries of research, the deeper mystery remains unsolved. Neuroscience can trace signals moving through neurons. It can observe brain activity lighting up during thoughts or emotions. It can even predict certain decisions before a person becomes aware of making them. But there is still one question that science has not fully answered. Why does any of this activity feel like something from the inside? Why does the firing of neurons produce experience? Tonight, we're going to take a calm, slow journey through the science that tries to answer this question. We'll explore how neurons communicate, how networks of cells turn signals into perception, how memory and emotion shape what we experience, and how the brain constructs the strange and deeply personal phenomenon we call the self. Along the way, we'll encounter surprising ideas from neuroscience, philosophy, and complex systems science, ideas that suggest the mind may be less like a machine and more like an emergent phenomenon, something that arises when a system becomes sufficiently complex and organized. And by the end of our journey, we may discover that the most extraordinary universe we know is not the one out among the stars, but the one quietly unfolding inside the human brain. So settle in, relax, and let your mind wander a little. Because tonight in the Midnight Lab, we're exploring the extraordinary story of how the brain creates a mind. Somewhere inside your skull, an extraordinary process is unfolding. A soft biological organ made of cells, water, fats, and proteins is producing a rich inner world filled with colours, sounds, memories, ideas, and emotions. From this living tissue arises the feeling of being someone, sitting somewhere, listening to a voice and reflecting on the nature of existence itself. That simple observation leads us directly into one of the deepest puzzles in all of science. How does matter become mind? Your brain is built from ordinary physical components. At its most basic level, it is made of biological cells communicating through electrical impulses and chemical signals. These signals move through networks that are constantly shifting, adapting, and reorganizing themselves as you learn, remember, and experience the world. And yet from this physical activity emerges something that feels entirely different from electricity and chemistry. It produces experience. It produces awareness. It produces the strange and unmistakable sensation that there is a self inside your head watching the world unfold. For thousands of years, thinkers have tried to understand how this transformation happens. One of the most influential early attempts came from the 17th century philosopher René Descartes. Writing in the early 1600s, Descartes proposed a bold idea that shaped centuries of debate. He suggested that the mind and the body were fundamentally different kinds of things. The body, according to Descartes, was physical and mechanical. It followed the laws of matter and motion, much like a clock or a machine. The mind, however, was something entirely different. Descartes believed the mind was a non-physical substance capable of thought, imagination, and awareness. In his famous formulation, the mind was the seat of consciousness, while the body was simply the machine that carried it around. This idea became known as mind-body dualism. In Descartes' view, the mind and the body interacted with each other, but they were not made of the same substance. Matter followed physical laws. Mind belonged to a different category of existence altogether. At the time, this idea seemed reasonable. After all, thoughts and feelings do not look or behave like physical objects. You cannot weigh a memory on a scale or measure the shape of a thought with a ruler. But as science advanced, this dualistic picture began to face serious challenges. Over the past 2 centuries, discoveries in biology, chemistry, and neuroscience have revealed something remarkable. Every time researchers look closely at mental processes, they find that these experiences correspond to measurable activity in the brain. Changes in neural circuits alter perception. Injuries to specific brain regions can disrupt language, memory, or emotion. Electrical stimulation of certain neural pathways can trigger vivid sensations or even complex memories. Again and again, evidence has pointed toward a powerful conclusion. Mental life appears to depend on the physical activity of the brain. This perspective led many scientists and philosophers to adopt a different approach. Instead of treating the mind as a separate substance, they began to ask whether consciousness might emerge naturally from biological processes. One of the most influential modern defenders of this view is philosopher John Searle. Searle argues for a position known as biological naturalism. According to Searle, consciousness is not something mystical or supernatural. It is a natural biological phenomenon produced by the brain in the same way that digestion is produced by the stomach or photosynthesis is produced by plants. The brain, in this view, generates conscious experience through the organized activity of its neural networks. But acknowledging that the brain produces consciousness does not actually solve the mystery. It simply reframes the question. Because now we must ask something even more puzzling. Why does neural activity feel like anything at all? In the 1990s, philosopher David Chalmers gave this puzzle a name that has since become famous. He called it the hard problem of consciousness. The hard problem asks why physical processes inside the brain give rise to subjective experience. We understand many aspects of brain function. Neuroscientists can explain how neurons transmit signals. They can map the networks involved in vision, hearing, and movement. They can identify brain regions that participate in memory, decision-making, and emotional responses. These are sometimes called the easy problems of consciousness, not because they are actually easy, but because they involve explaining functions and mechanisms. The hard problem is different. It asks why these mechanisms produce experience from the inside. Why does the firing of neurons produce the sensation of seeing the color red? Why does a particular pattern of brain activity feel like sadness, curiosity, or joy? Why does any of it feel like anything at all? Some researchers argue that answering this question requires combining philosophy and neuroscience in a more integrated way. One of the most prominent figures pursuing this approach is philosopher Patricia Churchland. Churchland helped pioneer the field known as neurophilosophy. Neurophilosophy explores philosophical questions about the mind using evidence from neuroscience. Instead of treating philosophy and brain science as separate disciplines, Churchland argues that understanding consciousness requires cooperation between them. Philosophers can clarify concepts and questions. Neuroscientists can investigate the physical systems that generate mental states. Together, these perspectives may gradually reveal how subjective experience emerges from neural processes, but even this partnership may not tell the entire story. Some scientists believe the answer may lie in a deeper principle that appears throughout nature: complexity. Across many scientific fields, researchers have discovered that when large numbers of simple components interact, entirely new properties can appear. These properties cannot be predicted simply by examining the individual parts in isolation. Water provides a familiar example. A single water molecule is not wet. Wetness emerges only when enormous numbers of molecules interact in liquid form. Similarly, individual neurons do not think or imagine, but when billions of them interact through intricate networks, something extraordinary seems to arise. Mind. One scientist who has explored this idea extensively is theoretical biologist Stuart Kauffman. Kauffman studies complex systems and the ways in which order and structure can emerge spontaneously in biological networks. His research suggests that when systems become sufficiently complex and interconnected, they may generate entirely new levels of organization. Life itself is one such emergent phenomenon, and the mind may be another. In this view, consciousness is not hidden inside any single neuron. It does not reside in one specific location within the brain. Instead, it may emerge from the collective dynamics of enormous networks of interacting cells. Billions of neurons exchanging signals through trillions of connections, patterns of activity constantly shifting, synchronizing and reorganizing themselves. A living system of extraordinary complexity. Somewhere within this vast web of electrical and chemical activity, the phenomenon we call mind appears. The feeling of being here, the awareness of a world around you, the quiet stream of thoughts passing through your mind as you listen to this moment. The ancient question remains open, but science has begun to illuminate the path toward understanding it. And that path leads us deeper into the brain itself, where the smallest components of the nervous system are carrying out an endless conversation. A conversation written in electricity, chemistry, and time. From those signals, somehow, the universe inside your mind begins to take shape. Inside your skull sits one of the most complex living systems known in the natural world. It weighs a little over 1 kilogram in most adults, has a texture not unlike soft tofu, and consumes roughly 20% of the energy your body produces, even though it represents only about 2% of your body's mass. That energetic appetite alone gives us an early hint that something extraordinary is happening inside. The human brain contains approximately 86 billion neurons. That number comes from careful research conducted by neuroscientist Susanna Herculana Housel, whose work in the early 2000s refined earlier estimates that had long circulated in neuroscience. Rather than relying on rough approximations, she used a method known as the isotropic fractionator, which allows researchers to count brain cells by dissolving tissue and analyzing the nuclei of individual cells. It sounds slightly destructive, but scientifically it is remarkably precise. Her findings revealed something striking. The human brain contains tens of billions of neurons, each capable of forming thousands of connections with others. When these connections are counted, the number of possible communication pathways rises into the trillions. That means that inside your head exists a network whose complexity rivals, and in some ways exceeds, many of the largest technological systems humans have ever built. But simply knowing the number of neurons does not explain how the brain works. If neurons were just isolated components sitting quietly beside one another, nothing very interesting would happen. The true power of the brain lies not in the individual cells themselves, but in the constantly changing patterns of interaction among them. To understand this, many neuroscientists describe the brain not as a machine with fixed parts, but as a dynamical system. The idea of a dynamical system comes from physics and mathematics. A dynamical system is any system whose state changes over time according to interacting processes. Weather systems are dynamical. Ocean currents are dynamical. The motion of planets around stars forms another example. In the late 20th century, neuroscientist Walter Freeman began applying this concept to the brain. Freeman studied how patterns of neural activity evolved moment by moment across large populations of neurons. His work suggested that brain activity behaves less like a rigid circuit and more like a constantly shifting landscape of electrical patterns. Rather than static signals traveling through fixed pathways, Freeman observed waves of activity moving through neural populations. These waves could change rapidly depending on sensory input, emotional state, or internal thought processes. In this view, the brain behaves like a living field of activity, continuously reorganizing itself in response to both the outside world and its own internal dynamics. One way this dynamic activity appears is through rhythmic patterns known as neural oscillations. Neural oscillations are repeating patterns of electrical activity that sweep across groups of neurons. These rhythms occur at different speeds, ranging from slow waves that cycle only a few times per second to very fast oscillations occurring dozens or even hundreds of times within the same second. Neuroscientist Georgi Buzaki has spent decades studying these rhythms and their role in brain function. His research has shown that neural oscillations are not random background noise. Instead, they appear to coordinate communication between different brain regions. Imagine a large orchestra preparing to perform. If every musician began playing at a completely different time, the result would be chaos. But when the musicians follow the same rhythm, the sounds combine into coherent music. Neural oscillations may serve a similar purpose in the brain. By synchronizing activity across different groups of neurons, rhythmic patterns can help coordinate the flow of information between distant brain regions. Different oscillation frequencies appear to play different roles. For example, slow rhythms often dominate during deep sleep, while faster rhythms become more prominent during focused attention and active thinking. These rhythmic patterns form part of the brain's internal timing system, organizing when neurons communicate with one another. Yet the brain's dynamics are not simply orderly rhythms marching along like a metronome. In fact, some scientists believe the brain operates in a delicate balance between order and disorder. This idea is captured by the concept of criticality. Criticality describes a state in which a system operates near a phase transition, balanced between stability and chaos. Physicists first studied this phenomenon in systems such as magnets and fluids, where small changes can produce large, unpredictable effects when a system approaches a critical point. Neuroscientist Dante Chialvo has explored the possibility that the brain operates near this critical state. His research suggests that neural networks may naturally organize themselves close to the boundary between rigid order and uncontrolled randomness. Why might this be useful? Because systems operating near criticality often display remarkable flexibility. They can respond quickly to new inputs, explore a wide range of possible activity patterns, and adapt rapidly to changing conditions. In a brain operating near criticality, a small signal from one neuron might spread across a network, triggering a cascade of activity that recruits many other neurons. At the same time, the system remains stable enough to prevent those cascades from spiraling completely out of control. This balance between order and chaos may allow the brain to remain both stable and creative, stable enough to maintain coherent perception, yet flexible enough to generate new ideas, learn from experience, and adapt to unfamiliar situations. Understanding such a complex system requires tools that go beyond traditional laboratory experiments. In recent years, researchers have begun using powerful computer models to simulate the brain's large-scale dynamics. One ambitious effort in this direction is known as the Virtual Brain Project. This international research initiative brings together neuroscientists, mathematicians, and computer scientists to create detailed digital simulations of brain activity. Using brain imaging data collected from real individuals, researchers construct computational models that approximate how signals travel across different brain regions. These simulations allow scientists to observe how patterns of activity evolve over time across the entire brain. Although these models are still developing, they offer a fascinating glimpse into the brain as a dynamic network rather than a collection of isolated parts. And this brings us to a crucial principle that shapes modern neuroscience. Mental processes do not arise from single neurons acting alone. No single neuron contains a thought. No individual brain cell stores an entire memory or emotion. Instead, mental phenomena emerge from coordinated activity across enormous populations of neurons. Perception, memory, imagination, and decision-making all depend on the collective behavior of networks that span multiple brain regions. Signals move through these networks in complex patterns, with activity rising and falling in different areas depending on the task at hand. In this sense, the brain behaves less like a traditional machine with fixed components, and more like a living ecosystem of electrical activity. Billions of cells constantly communicating, rhythms synchronizing distant regions, networks shifting their patterns as new information arrives. And from this constantly evolving dynamical system emerges the remarkable phenomenon we call the mind. The quiet awareness of the present moment, the ability to remember the past, the capacity to imagine futures that have not yet happened. all arising from the coordinated dance of neurons inside a living brain. At the foundation of everything the brain does lies a remarkable type of cell known as the neuron. If the brain is a vast living network, then neurons are the individual units that make the entire system possible. They receive information, process signals, and pass those signals onward to other cells in a continuous flow of communication. A single neuron is small, often only a fraction of a millimeter across, yet it is designed with extraordinary sophistication. Its structure reveals the basic strategy the brain uses to handle information. Most neurons have three main components. First is the cell body, sometimes called the soma. This is the central part of the neuron where the nucleus resides and where the cell maintains its basic biological functions. Branching outward from the cell body are thin extensions called dendrites. These delicate structures spread out like the branches of a tree, reaching toward neighboring neurons. Dendrites receive incoming signals from other cells, collecting information from thousands of potential sources. Then there is the axon. The axon is a long cable-like extension that carries signals away from the neuron toward other cells. In some neurons, the axon is only a short distance long. In others, it can extend many centimeters, traveling from the brain all the way down the spinal cord. Through this combination of dendrites and axon, neurons form a network capable of transmitting information across the entire nervous system. But how exactly does a neuron send a signal? The answer lies in one of the most important discoveries in modern neuroscience, the action potential. In the early 1950s, scientists Alan Hodgkin and Andrew Huxley conducted a series of groundbreaking experiments studying electrical activity in nerve cells. Working with the giant axon of a squid, which was large enough to insert delicate recording instruments into, they carefully measured how electrical signals travel along neurons. Their experiments revealed that neurons communicate through rapid electrical pulses generated by the movement of charged particles called ions across the cell membrane. At rest, a neuron maintains a difference in electrical charge between the inside of the cell and the surrounding fluid. The inside is slightly more negative than the outside, creating what scientists call the resting membrane potential. When the neuron receives enough incoming signals from other cells, this electrical balance changes. Specialized protein channels embedded in the cell membrane open, allowing sodium ions to rush into the neuron. This sudden movement of positively charged ions causes the electrical potential of the cell to rapidly shift. If the signal reaches a certain threshold, a dramatic electrical spike occurs. This spike is the action potential. Once triggered, the action potential travels down the axon like a wave moving along a wire. As it progresses, additional ion channels open in sequence, allowing the signal to propagate along the length of the neuron. Eventually, the electrical pulse reaches the end of the axon, where it triggers the release of chemical messengers that communicate with the next neuron in the network. Hodgkin and Huxley's work provided the first detailed mathematical description of how these electrical signals arise. Their model described the flow of ions across the cell membrane, using equations that explained how voltage changes over time. The core principle they uncovered can be summarized as follows. The rate of change of voltage across the neuron membrane equals the electrical current flowing through ion channels minus the current stored in the membrane's capacitance. Their research earned them the Nobel Prize in 1963 and laid the foundation for modern cellular neuroscience. For many years, scientists believed neurons operated somewhat like simple switches. A neuron would collect signals from its dendrites, add them together, and then either fire an action potential or remain silent. But more recent research has revealed that neurons are far more sophisticated than this simple picture suggests. One scientist who has explored this complexity in depth is computational neuroscientist Eden Segev. His work has focused on the internal structure of dendrites, those branching extensions that receive incoming signals. For a long time, dendrites were thought to function mainly as passive receivers of electrical input. They gathered signals from other neurons and passed them toward the cell body, where the final decision to fire an action potential would be made. However, Segev and his colleagues discovered that dendrites themselves contain a rich collection of ion channels and biochemical mechanisms. These structures allow dendrites to actively process information before it even reaches the main body of the neuron. In effect, dendrites can perform their own local computations. They can amplify certain signals, suppress others, and even generate localized electrical events within specific branches. This discovery suggests that neurons are not simply single processing units. Instead, each neuron may function as a miniature computational network, with different branches performing different operations simultaneously. Some neuroscientists now estimate that the computational capacity of a single neuron may be closer to that of a small neural network, rather than a simple on or off switch. When this idea is extended across the entire brain, the implications become staggering. Remember that the human brain contains roughly 86 billion neurons. If each of those neurons contains its own intricate internal processing capabilities, then the brain's overall computational complexity becomes almost unimaginably large. Christoph Koch, a neuroscientist known for his work on the neural basis of consciousness, has often emphasized that understanding the brain requires studying the circuits formed by neurons rather than focusing on individual cells alone. Koch has spent decades investigating how specific neural circuits contribute to conscious perception. His research, along with that of many other neuroscientists, suggests that conscious experience emerges from coordinated patterns of activity across networks of neurons. These networks may span multiple regions of the brain, linking sensory areas, memory systems, and decision-making circuits into unified patterns of activity. When you see a color, recognize a face, recall a memory, or form a thought, billions of neurons participate in that process. Electrical signals ripple across neural networks, chemical messengers are released between synapses, and rhythmic oscillations synchronize activity between distant brain regions. No single neuron contains the experience. Instead, the experience emerges from the collective behavior of the entire system. In this sense, neurons are the living circuitry of the brain. Each one is a microscopic processor capable of receiving signals, performing local computations, and transmitting information onward through the network. But it is the interaction among these countless cells that creates something far greater than the sum of its parts. Billions of neurons exchanging electrical signals, trillions of synaptic connections linking them together, patterns of activity shifting from moment to moment as the brain responds to the world and to its own internal dynamics. From this immense network arises the physical foundation of perception, memory, emotion, and thought. And somewhere within that constantly changing web of signals, the phenomenon we call the mind begins to appear. Electrical signals traveling through neurons may seem like the main language of the brain, but electricity alone does not tell the whole story. When one neuron wishes to pass information to another, it does not usually connect directly like two wires touching. Instead, the signal must cross a tiny gap. That microscopic space is called a synapse. A synapse is a specialized junction where communication between neurons takes place through chemistry. When an electrical impulse known as an action potential reaches the end of a neuron's axon, it triggers the release of molecules called neurotransmitters. These molecules drift across the narrow synaptic gap and bind to receptors on the neighboring neuron. When the receptors detect these molecules, they trigger electrical changes in the receiving cell. This chemical relay system is how information moves from one neuron to the next across the vast network of the brain. The discovery of this process marked a turning point in neuroscience, and much of the credit belongs to the Spanish neuroscientist Santiago Ramón y Cajal. In the late 1800s, Ramón y Cajal used an ingenious staining method, originally developed by the Italian scientist Camillo Golgi. The method involved treating brain tissue with silver salts, which randomly stained a small number of neurons in dark black against a lighter background. This allowed individual neurons to stand out clearly under a microscope. When Ramón y Cajal examined these stained samples, he noticed something remarkable. Instead of forming a continuous web, as many scientists had previously believed, neurons appeared to be separate cells. They approached each other closely but did not merge together. From these observations, Ramón y Cajal proposed what became known as the neuron doctrine. According to this principle, the nervous system is composed of individual cells that communicate with one another across specialized junctions. These junctions were later named synapses, and they form the fundamental communication points of the nervous system. Within a single human brain, there may be roughly 100 trillion synapses linking neurons together. Each synapse is capable of adjusting its strength over time, becoming more or less responsive depending on patterns of activity. This adaptability allows neural networks to change with experience, forming the biological basis of learning and memory. The chemicals that carry signals across synapses are known as neurotransmitters. Different neurotransmitters influence neurons in different ways. Glutamate is the most common excitatory neurotransmitter in the brain. When it binds to receptors on the receiving neuron, it increases the likelihood that the neuron will fire its own electrical signal. Gamma-aminobutyric acid, often abbreviated as GABA, works in the opposite direction. It is the main inhibitory neurotransmitter, decreasing the chance that a neuron will become active. But the brain's chemical language extends far beyond these basic excitatory and inhibitory signals. Some neurotransmitters act as broad regulators of brain activity. These substances are often called neuromodulators because they influence the overall behavior of large neural networks. Dopamine is one of the most widely studied examples. Dopamine plays a crucial role in motivation, learning, and reward. When an action leads to a positive outcome, dopamine signals help strengthen the neural pathways that produce that behavior. Serotonin, another well-known neuromodulator, influences mood, emotional stability, sleep cycles, and appetite. Changes in serotonin signaling are linked to mood disorders such as depression. These chemicals do not simply pass messages between two neighboring neurons. Instead, they often adjust the responsiveness of entire networks, subtly shifting the balance of activity across broad regions of the brain. For many years, neuroscientists believed neurons were the only cells actively involved in this communication process. Other cells found in the brain were considered supporting players at best. These supporting cells are known as glial cells, a name that comes from the Greek word for glue. Early researchers assumed glia existed mainly to hold neurons in place and provide structural support. But this assumption turned out to be far too modest. In recent decades, researchers have discovered that glial cells play surprisingly active roles in neural communication. Among the scientists who helped reveal this hidden layer of brain activity is neuroscientist Maiken Nedergaard. Nedergaard's research has focused on a type of glial cell called the astrocyte. Astrocytes are star-shaped cells that surround synapses and blood vessels throughout the brain. For a long time, they were thought to function primarily as maintenance workers, clearing away excess chemicals and supplying nutrients to neurons. However, experiments began to show that astrocytes respond dynamically to neural activity. When nearby neurons release neurotransmitters, astrocytes detect these signals and can release their own chemical messengers in response. This means astrocytes participate directly in the signaling process. They influence how synapses function, regulate the chemical environment around neurons, and help coordinate communication across networks. In fact, some researchers now describe the interaction between neurons and astrocytes as forming a tripartite synapse. Instead of communication occurring only between two neurons, a third participant, the astrocyte, helps shape the signal. The chemical complexity of the brain becomes even more fascinating when we consider a class of signaling molecules known as neuropeptides. Neuropeptides are small chains of amino acids that function as chemical messengers. Unlike many classical neurotransmitters, which act quickly and briefly, neuropeptides often produce slower but longer-lasting effects. One scientist who helped illuminate the importance of these molecules was Candace Pert. During the 1970s, Pert and her colleagues studied receptors for substances called endogenous opioids, naturally occurring chemicals in the brain that resemble opiate drugs. Their work revealed that neuropeptides are distributed throughout both the brain and the body. These molecules influence emotional states, pain perception, stress responses, and immune function. Pert proposed that neuropeptides help form a biochemical bridge between brain and body, linking emotional experiences with physical states. Another layer of complexity arises from the way chemical signals move through the brain's environment. For many years, neuroscientists focused primarily on direct synaptic communication, where neurotransmitters cross a tiny gap from one neuron to the next. But research led by neuroscientist Kjell Fiers introduced the concept of volume transmission. Volume transmission describes a process in which signaling molecules diffuse through the extracellular fluid that surrounds neurons. Instead of affecting only a single neighboring cell, these molecules can spread more broadly through brain tissue. This allows chemical signals to influence entire neighborhoods of neurons at once. Imagine a drop of dye placed into a glass of water. Instead of remaining confined to one small point, the dye gradually spreads outward, tinting the surrounding fluid. In a similar way, certain neurotransmitters and neuromodulators diffuse through brain tissue, shaping the activity of many nearby neurons simultaneously. Volume transmission provides yet another mechanism through which the brain coordinates its immense networks. When we step back and consider all these processes together, a remarkable picture begins to emerge. Neurons send electrical impulses along their axons. At synapses, chemical neurotransmitters carry signals across microscopic gaps. Neuromodulators adjust the responsiveness of entire neural systems. Astrocytes participate in signaling, influencing synaptic activity and regulating the chemical environment. Neuropeptides connect emotional states with bodily physiology. And volume transmission allows chemical signals to diffuse through brain tissue, influencing networks of cells beyond individual synapses. The brain's communication system is therefore not simply electrical and not purely chemical. It is an intricate hybrid of both, operating across multiple spatial scales and timescales. Within this vast chemical conversation, billions of neurons exchange signals every moment. And from the shifting patterns of that communication, the dynamic activity of the mind continues to unfold. If a single neuron were responsible for producing a thought, understanding the brain would be relatively straightforward. We could simply locate the neuron that generates a memory, isolate the one that produces joy, perhaps even identify the exact cell responsible for curiosity. But the brain does not operate that way. Instead, every thought, perception, and memory arises from the activity of large networks of neurons interacting with one another. These networks extend across many regions of the brain, linking billions of cells into a constantly shifting system of communication. To understand how these enormous systems function, neuroscientists increasingly borrow tools from another field of science known as network theory. Network theory studies how complex systems are organized when many individual units are connected together. It has been used to analyze systems as varied as airline transportation routes, social media connections, power grids, and even patterns of collaboration among scientists. In the early 2000s, neuroscientist Olaf Sporns began applying these ideas to the brain. Sporns helped establish a field now known as network neuroscience, which treats the brain as a complex graph network composed of nodes and connections. In this framework, individual brain regions or neurons can be thought of as nodes, while the connections between them are called edges. By mapping these connections and studying their patterns, researchers can begin to understand how information flows through the brain. When scientists first started analyzing brain connectivity in this way, they discovered something surprising. The brain's network structure resembles a type of architecture known as a small world network. The concept of small world networks was introduced by mathematicians Duncan Watts and Stephen Strogatz in the late 1990s. They were studying systems in which most nodes are connected to nearby neighbors, while a smaller number of long-range connections link distant parts of the network. This combination produces a network with two important properties. First, clusters of nodes form tightly interconnected local groups. Within these clusters, information can move quickly between neighboring nodes. Second, a smaller number of long-range connections bridge distant clusters, allowing signals to travel efficiently across the entire network. The result is a system that combines local specialization with global integration. In everyday life, a similar pattern appears in social networks. Most people have close relationships with family members, co-workers, or neighbors who live nearby. At the same time, occasional connections to people in distant cities or countries link these local communities together. Because of those long-range connections, information can spread rapidly across the entire network, even when most relationships remain local. The brain appears to follow this same organizational principle. Neurons tend to form dense clusters with nearby cells, creating local circuits that specialize in particular functions. For example, certain regions of the visual cortex process specific features of visual input such as edges, motion, or color. At the same time, long-range neural pathways connect distant brain regions. These connections allow information from the visual system to interact with memory systems in the temporal lobe, emotional processing circuits in the limbic system, and decision-making networks in the frontal cortex. Because of this architecture, the brain can process information both locally and globally at the same time. But the brain's network organization contains another intriguing feature known as rich club connectivity. In many complex networks, certain nodes become far more highly connected than others. These nodes act as hubs, coordinating communication across different parts of the system. Researchers studying brain connectivity discovered that some brain regions form a densely interconnected core network of hubs. These highly connected regions preferentially connect with one another, forming what scientists call a rich club. The term may sound slightly playful, but the idea is scientifically important. Rich club connectivity refers to the tendency of the most connected nodes in a network to link directly with each other. In the brain, these hub regions play crucial roles in coordinating information flow between specialized processing areas. For example, regions within the frontal cortex, parietal cortex, and certain deep brain structures often serve as central hubs. These areas integrate information from sensory systems, memory networks, and emotional circuits. Because these hubs connect to many different parts of the brain, they are ideally positioned to combine diverse streams of information into coherent mental processes. If one of these hubs becomes damaged through injury or disease, the consequences can be surprisingly widespread. Disrupting a central hub can interrupt communication across large portions of the brain's network, leading to significant cognitive difficulties. Understanding these patterns of connectivity has led neuroscientists to pursue an ambitious goal: mapping the complete wiring diagram of the brain. This idea is known as the connectomy. The connectome represents the full map of neural connections within a nervous system. Just as the human genome maps the sequence of genes within our DNA, the connectome seeks to map the structure of connections linking neurons together. One of the scientists who has strongly advocated for this approach is neuroscientist Sebastian Seung. Seung has argued that the patterns of connections between neurons may play a fundamental role in shaping who we are. In his view, memories, skills, and even aspects of personality may be encoded within the structure of neural connections. If those connections change through learning or experience, the organization of the brain changes as well. Seung famously summarized this idea with a simple phrase: "You are your connectome." While this statement simplifies a complex topic, it captures an important insight. The wiring patterns linking neurons together help determine how information flows through the brain. And because those patterns evolve throughout life, they contribute to the unique mental landscape of every individual. Mapping these connections at full resolution, however, is an enormous technical challenge. The human brain contains trillions of synaptic connections, each smaller than the width of a human hair by many orders of magnitude. To address this complexity, researchers have begun using powerful computer simulations to model small sections of brain circuitry in extraordinary detail. One of the most ambitious efforts in this direction is the Blue Brain Project. Led by neuroscientist Henry Markram and based at the Swiss Federal Institute of Technology in Lausanne, the Blue Brain Project aims to build digital simulations of cortical microcircuits. The project uses experimental data collected from biological neurons to construct detailed computer models of neural networks. These models attempt to replicate how groups of neurons interact through electrical and chemical signaling. By simulating these circuits on supercomputers, researchers can explore how patterns of activity emerge from the interactions between individual neurons. Although these simulations currently represent only tiny fractions of the full brain, they offer valuable insights into how complex neural dynamics arise. The broader lesson emerging from all of this research is both simple and profound. Mental phenomena do not reside in isolated locations within the brain. A memory is not stored in a single neuron. An emotion does not originate from a single circuit acting alone. Instead, mental processes arise from patterns of activity distributed across vast networks of interconnected neurons. These networks constantly reorganize themselves as you perceive the world, recall past experiences, and imagine future possibilities. Electrical signals ripple through the network. Chemical messengers adjust the strength of connections. Rhythmic oscillations synchronize distant brain regions. Through these coordinated interactions, the brain forms a dynamic system capable of extraordinary feats. Recognizing a familiar face in a crowd, understanding the meaning of a sentence, reflecting on your own thoughts as they unfold. Each of these experiences emerges from the coordinated activity of billions of neurons linked together in one of the most intricate networks known to science. From that immense web of connections, the phenomenon we call mind begins to take shape. At every moment, your brain is receiving information from the world around you. Light enters your eyes, vibrations reach your ears, pressure touches your skin, molecules drift through your nose, and chemical signals activate receptors on your tongue. Each of these events begins as a physical interaction between the environment and the body, yet the brain itself never directly experiences light, sound, or texture. Inside the skull, there is only electrical activity moving through networks of neurons. This means the brain must perform an extraordinary transformation. It must convert physical energy from the outside world into signals that neurons can understand. This transformation is known as sensory transduction. Sensory transduction is the process by which specialized cells in the body translate environmental stimuli into electrical activity within the nervous system. Without this process, the brain would remain completely isolated from the outside world, surrounded by darkness and silence despite everything happening beyond the skull. Consider vision as an example. Light from the environment enters the eye through the cornea and lens, eventually reaching the retina at the back of the eyeball. The retina contains specialized cells known as photoreceptors. These cells detect incoming light and convert it into electrical signals. There are two main types of photoreceptors. Rod cells are highly sensitive to light and allow us to see in dim conditions, such as during twilight or at night. Cone cells are less sensitive to low light but are responsible for detecting color and fine visual detail. When photons of light strike these cells, they trigger a chain of biochemical reactions that change the electrical properties of the cell membrane. This process ultimately produces electrical signals that travel along neurons connecting the retina to the brain. What began as electromagnetic radiation traveling through space has now become a neural signal moving through the nervous system. A similar transformation occurs in the sense of touch. Your skin contains specialized receptors known as mechanoreceptors. These cells respond to mechanical forces such as pressure, stretching, and vibration. When the skin is pressed or deformed, the membranes of these receptors change shape, opening tiny ion channels that allow electrically charged particles to flow into the cell. This flow of ions generates electrical signals that travel along sensory neurons toward the spinal cord and eventually the brain. Sound follows yet another path. Inside the inner ear lies the cochlea, a spiral-shaped structure filled with fluid. When sound waves enter the ear, they cause tiny vibrations in this fluid. These vibrations move delicate structures known as hair cells. Despite the name, hair cells are not actually hair. They are sensory receptors topped with bundles of microscopic filaments called stereotelia. As these structures bend in response to fluid movement, they open ion channels in the cell membrane. Once again, mechanical energy is converted into electrical activity. Within a fraction of a second, sound waves traveling through the air become patterns of neural signals moving through the auditory nerve toward the brain. Across all of these senses, the same principle applies. Physical energy is transformed into electrical activity within neurons. The brain receives these signals and uses them as raw material for perception. However, the brain does not receive these signals as simple copies of the world. In fact, much of the processing has already begun long before the signals reach the higher regions of the brain. Vision provides a particularly clear example of this early processing. The retina itself contains multiple layers of neurons that analyze visual information before it even leaves the eye. These retinal circuits detect contrasts, motion, and edges in the visual scene. By the time signals reach the brain through the optic nerve, they have already been partially processed. Further processing occurs in the visual cortex, a region of the brain located at the back of the head. In the 1950s and 1960s, neuroscientists David Hubel and Torsten Wiesel conducted a series of elegant experiments studying how neurons in the visual cortex respond to visual stimuli. Working with cats and monkeys, they recorded the activity of individual neurons while presenting patterns of light on a screen. They discovered that certain neurons responded specifically to edges oriented in particular directions. For example, one neuron might fire strongly when it detected a vertical line of light but remain quiet when the line was horizontal. Another neuron might respond to diagonal patterns or moving edges. Hubel and Wiesel concluded that visual perception arises through a hierarchy of specialized neurons, each tuned to detect particular features of the visual world. Some neurons detect simple edges, others detect motion or shape, and higher-level circuits combine these signals to recognize objects. Their discoveries earned them the Nobel Prize in 1981 and revealed that the brain is already performing sophisticated analysis of sensory input at very early stages of processing. These findings challenge a common intuition about perception. It might seem as though the brain functions like a camera, faithfully recording the external world exactly as it appears. But in reality, perception involves interpretation and inference from the very beginning. Even the earliest stages of sensory processing involve extracting patterns and organizing information. Psychologist James J. Gibson offered another influential perspective on this process. Gibson was an ecological psychologist who argued that perception should be understood in terms of the relationship between an organism and its environment. According to Gibson, perception is not simply about reconstructing the physical world inside the brain. Instead, it involves detecting opportunities for action within the environment. He introduced the concept of affordances. An affordance refers to the possibilities for action that objects and environments provide. A chair affords sitting, a staircase affords climbing, a path affords walking. From this viewpoint, the brain does not merely build an internal picture of the world, it interprets sensory information in terms of how the body can interact with its surroundings. Perception, therefore, becomes an active process shaped by both sensory input and behavioral goals. This idea highlights a deeper truth about the brain's relationship with the world. Perception is not passive recording. The brain is not a silent observer receiving perfect copies of external reality. Instead, it actively interprets the signals arriving from the senses, organizing them into meaningful patterns. Electrical signals from photoreceptors become visual scenes. Vibrations detected by hair cells become music and speech. Pressure on the skin becomes the sensation of touch. At every moment, the brain is transforming raw physical signals into the rich sensory world you experience. And all of it begins with the remarkable ability of sensory systems to translate the language of physics into the language of neurons. Take a moment to imagine something simple. A red apple sitting on a wooden table. You might notice the smooth curve of its shape, the deep red color of the skin, the faint reflection of light on its surface, perhaps even the expectation of its crisp taste, or the subtle scent that apples sometimes carry. All of these qualities seem to belong to one single object. Your mind experiences the apple as a unified thing, a single, coherent presence in the world. But inside your brain, the situation looks very different. The color red is processed in one group of neurons. The shape of the apple is analyzed by other neurons sensitive to edges and contours. Motion-detecting neurons would activate if the apple rolled across the table. Other circuits might evaluate whether the object is edible or familiar. Even the recognition that it is an apple, rather than simply a round red shape, involves additional memory networks interacting with the visual system. In other words, different features of the apple are processed in different parts of the brain. And this leads to a fascinating puzzle known in neuroscience as the binding problem. The binding problem asks a simple question that turns out to be remarkably difficult to answer. How does the brain combine separate streams of information into a single, unified experience? If color, shape, motion, location, and meaning are processed in different neural circuits, why do they appear together as one object in your perception? Why do you experience a single apple rather than a collection of disconnected sensory features floating independently through your mind? The brain solves this problem constantly, yet the mechanism behind it remains one of the most intriguing challenges in neuroscience. One influential idea comes from the German neuroscientist Wolf Singer. Singer proposed that the brain may bind sensory information together using synchronized neural activity. According to this hypothesis, neurons representing different features of the same object may fire in rhythmic synchrony. Imagine multiple groups of neurons scattered across the brain. One group encodes color, another encodes shape, and another encodes location. If these groups fire electrical signals at precisely the same rhythm, their activity may become linked as part of a single perceptual event. Singer suggested that synchronized oscillations in neural activity could act as a kind of timing signal, indicating which neurons belong together in the same perceptual representation. In this view, timing becomes just as important as location in the brain. Neurons representing the same object may fire together within the same rhythmic cycle, while neurons representing other objects fire at slightly different moments. This subtle coordination could help the brain organize multiple features into coherent experiences. The idea gained support from experiments showing that groups of neurons sometimes display synchronized oscillations during perception. In particular, rhythmic activity in frequency ranges known as gamma oscillations appears to increase when animals recognize objects or focus attention on specific stimuli. However, synchrony alone may not tell the entire story. Another neuroscientist, Pascal Fry, has explored a related concept known as neural coherence. Fries proposed that communication between brain regions depends on the alignment of rhythmic activity across those regions. When two groups of neurons oscillate in phase with each other, their signals can be transmitted more efficiently. If their rhythms fall out of sync, communication becomes less effective. Fries described this mechanism as communication through coherence. To understand the idea, imagine two people trying to have a conversation across a large room. If they speak at the same moment and listen at the same moment, their exchange flows smoothly. But if one person speaks while the other is not listening, the message is lost. Similarly, neurons appear to communicate most effectively when their oscillatory rhythms are aligned. When multiple brain regions synchronize their activity, they form temporary communication channels. These channels allow information to travel between areas that may be physically distant within the brain. Through this mechanism, the brain can dynamically link networks that represent different aspects of perception. For example, neurons detecting the color of an object might synchronize their activity with neurons analyzing its shape and location. When these rhythms align, the networks effectively join forces, allowing the brain to construct a unified representation of the object. The remarkable aspect of this process is that these connections are not permanent. Neural synchrony can change from moment to moment depending on what you're seeing, hearing, or thinking. Networks may link together briefly to represent a particular perception, then dissolve and reorganize as new information arrives. This flexibility allows the brain to bind different features together rapidly, while still maintaining the ability to represent many different objects at the same time. Without such a binding mechanism, perception would become chaotic. Imagine looking at a busy street scene where cars, people, buildings, and streetlights are all present. If the brain could not correctly bind features together, colors might drift away from shapes, movements might detach from the objects producing them, and sounds might become disconnected from their sources. Instead of a coherent world, you might experience a confusing collage of unrelated sensations. Fortunately, the brain's binding mechanisms appear to work remarkably well. Through coordinated activity across distributed networks, separate features are integrated into unified objects. Those objects then form scenes, and those scenes become the continuous flow of experience we call perception. The binding problem highlights something profound about the nature of the mind. Our conscious experience feels seamless and unified, yet it emerges from a system composed of billions of specialized components working in parallel. The brain does not rely on a single central processor, assembling all information in one place. Instead, unity arises from cooperation among many distributed networks, neurons communicating through electrical signals, rhythms synchronizing distant regions, networks forming temporary alliances as different features of the world are brought together. Through these coordinated processes, the brain transforms scattered pieces of sensory information into the coherent reality you experience. A single apple on a table, a friend's face across a room, the quiet awareness of your surroundings in this moment, all appearing as unified experiences within the vast and intricate network of the brain. When you open your eyes and look at the world, it may feel as though your brain is simply receiving information from the environment. Light enters the eyes, sound reaches the ears, and the brain quietly records what is happening around you. But a growing body of research suggests that the brain may be doing something far more active than simple observation. According to one influential framework in modern neuroscience, the brain is constantly making predictions about the world. Rather than passively waiting for sensory information to arrive, it is continuously generating expectations about what it is likely to see, hear, and feel next. Incoming signals from the senses are then compared against these expectations. This idea forms the basis of a theory known as predictive processing. One of the central figures behind this approach is neuroscientist Carl Friston. Friston has spent decades developing mathematical models that attempt to describe how biological systems maintain stability while interacting with complex environments. At the heart of his theory lies a concept called the free energy principle. Despite the name, this idea does not refer to energy in the everyday sense of fuel or electricity. Instead, free energy in this context refers to a mathematical measure of uncertainty or surprise within a system. Friston's proposal is that living systems, including the brain, constantly act to minimize this kind of surprise. In other words, the brain attempts to maintain internal models of the world that allow it to anticipate incoming sensory information. When those predictions are accurate, the system remains stable and efficient. When predictions fail, the brain must adjust its internal model to better match reality. The principle can be summarized in a simple way. The brain seeks to minimize the difference between predicted sensory signals and the signals that actually arrive from the environment. This difference is often called prediction error. Prediction error plays a crucial role in learning and perception. Whenever the brain encounters information that does not match its expectations, that mismatch generates a signal indicating that the internal model needs updating. Over time, the brain refines its predictions by incorporating these errors, gradually improving its ability to anticipate the structure of the world. Philosopher Andy Clark has explored the implications of this idea in depth. Clark describes the brain as a prediction machine that constantly forecasts the sensory consequences of events in the environment. Instead of constructing perception from the ground up using raw sensory data, the brain begins with hypotheses about what is likely to be present. Sensory input then acts as a stream of evidence that confirms or challenges those hypotheses. In this view, perception becomes a process of continuous hypothesis testing. The brain proposes a model of the world. Incoming sensory signals either support that model or reveal discrepancies. Those discrepancies generate prediction errors that drive learning and adjustment. Over time, the brain becomes better at anticipating the patterns of its environment. This framework leads to a rather surprising conclusion about the nature of perception. Much of what you experience may not be directly caused by incoming sensory signals. Instead, your brain may be actively generating much of your perceptual experience based on its expectations. Neuroscientist Anil Seth has described this idea using a memorable phrase. He suggests that perception functions as a controlled hallucination. The term hallucination may sound dramatic, but the meaning is more subtle than it first appears. A hallucination normally refers to a perception that occurs without any external stimulus. But in Seth's description, perception is still guided by sensory input. The key difference is that the brain's internal predictions play a central role in shaping what you experience. Your brain generates a model of the world based on past experiences, learned patterns, and contextual information. Incoming sensory signals then modify this model by providing evidence about what is actually happening. Perception therefore arises from a combination of two processes: predictions flowing downward from the brain's internal models, sensory signals flowing upward from the sense organs. Where these two streams meet, perception emerges. This process can be observed in many everyday situations. Consider reading a sentence in your native language. Your brain does not examine each individual letter in isolation. Instead, it predicts likely words and phrases based on context and experience. Even when letters are partially obscured or slightly misspelled, the brain often fills in the missing information automatically. Visual illusions provide another example. Certain images can cause the brain to perceive shapes or colors that are not physically present in the stimulus. These illusions reveal how strongly perception is influenced by expectations built into the brain's internal models. Prediction also plays an important role in action. When you reach out to pick up a cup, your brain predicts how your muscles should move and what sensory feedback should occur. If the cup suddenly shifts position or feels heavier than expected, the resulting prediction error allows your brain to adjust the movement almost instantly. This constant cycle of prediction and correction helps explain how the brain can operate efficiently in a complex and changing world. Instead of processing every sensory detail from scratch, the brain focuses its computational resources on unexpected information. Signals that match predictions require little additional processing. Signals that violate expectations trigger learning and adjustment. This strategy greatly reduces the amount of computation required to interpret sensory input. It also highlights the deep relationship between perception and experience. Your brain's predictions are shaped by memories, habits, cultural knowledge, and previous encounters with the world. Two individuals observing the same scene may therefore interpret it differently because their internal models are not identical. Perception becomes a collaboration between the world and the brain. External signals provide the raw evidence. Internal predictions provide the interpretive framework. When these two sources align, perception feels clear and stable. When they diverge, confusion or surprise arises, prompting the brain to revise its understanding. Over time, this continuous feedback process allows the brain to build increasingly sophisticated models of reality. The result is not a perfect reconstruction of the external world. Instead, it is a best guess, a dynamic interpretation produced by the brain as it balances expectation with incoming evidence. In this sense, every moment of perception represents a successful negotiation between the brain's predictions and the sensory signals arriving from the environment. Your experience of the world is not simply delivered to you by your senses. It is actively constructed by a brain that is constantly anticipating what will happen next. Every moment you experience becomes part of a continuous story unfolding inside the mind. The places you have visited, the people you have met, the conversations you remember, even the faint recollection of a smell from childhood, all form part of a vast internal archive. This archive is what we call memory. Without memory, experience would dissolve almost as quickly as it appeared. Each moment would fade before the next could build upon it. Learning would be impossible, relationships could not develop, and the sense of personal identity would vanish. Memory provides continuity. It allows the past to shape the present and gives structure to the unfolding stream of experience. One of the most important structures involved in forming memories is a small region of the brain called the hippocampus. The hippocampus sits deep within the temporal lobe on each side of the brain. Its name comes from the Greek words for horse and sea monster, because its curved shape reminded early anatomists of a tiny seahorse. Despite its modest size, the hippocampus plays a central role in organizing certain types of memory, particularly episodic memory. Episodic memory refers to the ability to recall specific events from one's personal life. Remembering where you parked your car this morning, recalling a conversation with a friend, or picturing the room where you're listening right now all rely on episodic memory. The importance of the hippocampus in forming these memories became dramatically clear through the story of a single patient whose experience changed the course of neuroscience. His name was Henry Molaison. In the early 1950s, Henry suffered from severe epilepsy that could not be controlled by medication. In an effort to reduce his seizures, surgeons performed an experimental operation in 1953. During the procedure, they removed large portions of his medial temporal lobes, including much of the hippocampus. The surgery successfully reduced his seizures, but it produced an unexpected and profound consequence. Henry lost the ability to form new episodic memories. After the operation, he could carry on a conversation and appear perfectly normal for several minutes. But if he left the room and returned shortly afterward, he would greet the same person as though meeting them for the first time. His memories of childhood and early life remained largely intact, but events that occurred after the surgery could not be stored in long-term memory. Henry Molaison became one of the most carefully studied patients in the history of neuroscience. For decades, researchers worked with him to understand how the brain forms and stores memories. His case revealed a crucial insight. The hippocampus is not simply a storage container for memories. Instead, it functions as a kind of organizer or index that helps encode new experiences so they can later be retrieved. Over time, memories appear to become distributed across networks of neurons throughout the cortex. The hippocampus plays a vital role during the early stages of this process, helping to link together the various elements of an experience. These elements might include visual details, sounds, spatial context, emotional responses, and the sequence of events that occurred. But how are memories physically represented inside the brain? Scientists have long suspected that memories correspond to specific patterns of neural activity. The idea that a memory leaves behind a physical trace in the brain is known as the engram. In recent decades, neuroscientist Susumu Tonegawa and his colleagues at the Massachusetts Institute of Technology have conducted experiments exploring how these memory traces form. Using techniques that allow researchers to identify and manipulate specific neurons in laboratory animals, Tonegawa's team discovered that particular groups of neurons become active when a memory is formed. These groups of neurons can later be reactivated when the memory is recalled. In some experiments, scientists even artificially stimulated these neurons to trigger the behavioral expression of a memory. When the engram cells associated with a particular experience were activated, animals behaved as though they were reliving that experience. These findings provide strong evidence that memories correspond to patterns of activity distributed across networks of neurons. Yet memory is not as stable as we might imagine. When we recall a memory, it does not simply play back like a recording stored in a digital archive. Instead, the act of remembering can actually alter the memory itself. This phenomenon is known as memory reconsolidation. When a memory is retrieved, it temporarily becomes unstable. During this period, new information can be incorporated into the memory before it is stored again. As a result, memories may gradually change over time as they are repeatedly recalled and updated. This explains why people sometimes remember events differently after many years have passed. Each act of remembering can subtly reshape the memory. Rather than being perfectly preserved snapshots of the past, memories behave more like living documents that evolve with each retrieval. The dynamic nature of memory also connects closely with another remarkable ability of the human mind: imagination. Psychologist Daniel Schacter and his colleagues have studied how the brain constructs both memories of the past and simulations of possible futures. Their research has revealed that many of the same brain regions are involved in both processes. When people recall past experiences, activity increases in networks that include the hippocampus, the medial prefrontal cortex, and parts of the parietal lobe. Interestingly, these same networks become active when people imagine events that have not yet happened. This suggests that the brain may use memory systems not only to store past experiences, but also to construct mental simulations of possible future situations. In other words, remembering and imagining rely on many of the same neural mechanisms. This ability allows the brain to do something extraordinary. It can use fragments of past experiences to construct new possibilities. A familiar place combined with a new scenario, a remembered face placed in an imagined conversation, a future plan built from elements of past events. Memory therefore serves not only as a record of what has happened, but also as a toolkit for exploring what might happen next. And perhaps most importantly, memory constantly shapes how we interpret the present. When you encounter a new situation, your brain automatically draws upon past experiences to guide perception and decision-making. Familiar patterns are recognized quickly because they resemble memories stored within neural networks. A particular smell might trigger recollections of childhood. A familiar melody may evoke emotions connected to earlier moments in life. Even subtle cues can activate memory systems that influence how we understand what is happening right now. In this sense, perception is never entirely separate from memory. The present moment is always filtered through the accumulated experiences of the past. Every memory stored within the brain becomes part of the architecture through which new experiences are interpreted. Through this ongoing interaction between past and present, memory helps shape the evolving narrative of the mind. Long before humans began writing poetry, building cities, or wondering about the nature of consciousness, the brain had already developed powerful systems for something much older. Emotion. Emotion is often treated as the opposite of reason, something messy that interferes with clear thinking. Yet from the perspective of neuroscience, emotions are not accidents or distractions. They are ancient guidance systems that evolved to help organisms survive in complex and unpredictable environments. Deep within the brain lie circuits that evaluate situations, generate feelings, and prepare the body for action. These systems influence attention, shape memories, guide decisions, and help determine what matters in the world around us. Two structures play especially important roles in these emotional processes. One is the amygdala. The amygdala is a small almond-shaped cluster of neurons located deep within the temporal lobe. Despite its modest size, it acts as an important hub for detecting emotionally significant events, especially those related to potential threats. When sensory information arrives in the brain, signals can reach the amygdala very quickly, sometimes even before more detailed analysis occurs in higher cortical regions. This rapid pathway allows the brain to respond to possible dangers with remarkable speed. For example, if a sudden movement appears in the corner of your vision, or a loud noise echoes unexpectedly behind you, the amygdala can trigger a cascade of physiological responses almost immediately. Heart rate increases, attention sharpens, muscles prepare for action. The body becomes ready either to confront the situation or to move away from it. Another key structure involved in emotional regulation is the hypothalamus. Located near the base of the brain, the hypothalamus acts as a bridge between the nervous system and the endocrine system. When emotional circuits detect something significant, the hypothalamus can initiate hormonal responses that influence the entire body. It communicates with the pituitary gland, which then releases hormones into the bloodstream affecting organs throughout the body. This system helps coordinate physiological states associated with emotion, such as the release of stress hormones during danger, or the calming shifts that accompany safety and relaxation. These emotional circuits are not unique to humans. In fact, many of them are shared across a wide range of mammals. Neuroscientist Jacques Panksepp spent much of his career studying these ancient emotional systems. Through experiments with animals, Panksepp identified several fundamental emotional circuits that appear to be deeply rooted in mammalian brains. He proposed that mammals share a set of primary emotional systems, including seeking, fear, rage, lust, care, panic, and play. Each of these systems corresponds to neural circuits that guide behavior in specific contexts. The seeking system, for example, drives exploration and curiosity. It motivates animals to search for food, investigate new environments, and pursue goals. The fear system prepares the organism to avoid danger. The care system supports nurturing behavior, particularly between parents and offspring. The play system encourages social interaction and learning among young animals. Panksepp's research suggested that these emotional systems form a foundational layer of the brain's motivational architecture. They shape behavior long before complex reasoning enters the picture. In this sense, emotions provide a kind of internal compass, helping organisms navigate the challenges of survival. But emotions do more than prepare the body for action. They also influence how decisions are made. Neurologist Antonio Damasio explored this connection through a theory known as the somatic marker hypothesis. Damasio studied patients who had damage to parts of the frontal cortex involved in emotional processing. Surprisingly, many of these individuals retained their logical reasoning abilities. They could analyze situations, weigh options, and explain potential outcomes. Yet they often struggled to make everyday decisions. Without normal emotional signals, even simple choices became difficult. Patients might spend long periods deliberating over minor details, unable to determine which option felt preferable. Damasio proposed that emotions provide signals that help guide decision-making. According to the somatic marker hypothesis, emotional responses associated with past experiences become linked to particular outcomes. When a similar situation arises in the future, these emotional markers influence the decision process. For instance, if a previous choice led to negative consequences, the memory of that event may trigger an uneasy feeling when a similar situation appears again. That emotional signal acts as a warning, steering behavior away from repeating the mistake. Conversely, positive emotional markers may encourage actions that previously led to rewarding outcomes. In this way, emotions help narrow the range of possible choices, allowing decisions to be made more efficiently. More recently, neuroscientist Lisa Feldman Barrett has proposed a different perspective on how emotions arise. Barrett's theory, known as the theory of constructed emotion, suggests that emotions are not fixed biological programs triggered automatically by specific stimuli. Instead, she argues that emotions are constructed by the brain through predictive processes. According to this view, the brain continuously interprets signals coming from the body and the environment. Using past experiences and cultural knowledge, it predicts what emotional state might best explain the current situation. For example, an increased heart rate could indicate excitement, fear, anticipation, or physical exertion. The brain interprets these bodily signals within a particular context and constructs an emotional experience that fits the situation. In Barrett's framework, emotions are not universal packages hardwired into the brain. They are dynamic interpretations shaped by learning, memory, and prediction. Although this perspective differs from earlier theories, it still emphasizes a central point. Emotion and cognition are deeply intertwined. Emotional signals influence what we notice, how we interpret events, and what we remember later. A surprising event accompanied by strong emotion is often remembered far more vividly than an ordinary moment. Likewise, emotional states can shape perception itself. When someone feels anxious, the brain may become more sensitive to potential threats in the environment. When someone feels relaxed and safe, attention may shift toward exploration and curiosity. Emotions also influence motivation. They help determine what goals feel important and which actions seem worth pursuing. Rather than existing separately from thinking, emotions help guide the entire cognitive system. They influence perception, they shape memory, they guide action, and they provide a powerful motivational framework that helps organisms navigate the complexities of the world. What might sometimes feel like irrational impulses are in fact the echoes of ancient neural systems that have been refined over millions of years of evolution. Systems designed not merely to experience the world, but to survive within it. Within the living network of the brain, emotion serves as both signal and guide, quietly shaping the choices, perceptions, and memories that make up the unfolding story of the mind. When people imagine the mind, they often picture something happening entirely inside the brain. Thoughts appear to arise from neural activity within the skull, and it is tempting to assume that cognition is simply the product of this isolated organ. But a growing body of research suggests that this picture may be incomplete. The brain does not operate alone. Instead, the mind appears to emerge from a continuous interaction between the brain, the body, and the surrounding environment. Neural activity is deeply intertwined with the signals flowing through the body's organs, muscles, hormones, and sensory systems. In other words, the mind may be embodied. One of the thinkers who helped develop this perspective was neuroscientist and philosopher Francisco Varela. Varela argued that cognition cannot be fully understood if we treat the brain as a detached information processor. Rather than functioning like a computer sealed inside the skull, the brain is part of a living system that constantly interacts with the world through the body. This view became known as embodied cognition. Embodied cognition proposes that many aspects of thinking arise from the body's physical interactions with its environment. The way we perceive space, manipulate objects, and move through the world shapes the structure of our cognitive processes. Consider something as simple as understanding the concept of balance. Our comprehension of balance is not merely abstract. It is rooted in bodily experiences such as standing upright, walking across uneven ground, or steadying ourselves when the surface beneath us shifts. Similarly, our understanding of direction, distance, and movement relies on sensory signals from muscles, joints, and the inner ear. These bodily experiences become the foundation upon which more abstract forms of reasoning are built. Varela extended these ideas further through a framework called enactivism, developed together with philosopher Evan Thompson and psychologist Eleanor Roche. Enactivism proposes that cognition emerges through active engagement with the world. The mind does not passively represent an external reality. Instead, it actively participates in shaping experience through perception and action. In this framework, the brain does not simply construct internal models of a pre-existing world. Rather, organisms bring forth meaningful worlds through their interactions with the environment. For example, a forest means different things to different creatures. To a bird, it may be a network of branches for nesting and navigation. To a deer, it may be a landscape of shelter and food. To a human hiker, it might be a place of exploration or reflection. Each organism experiences the environment through the lens of its own bodily capabilities and needs. This perspective emphasizes that cognition is always situated. The mind emerges through the continuous loop of perception and action, linking brain, body, and world. One of the most fascinating aspects of this interaction involves a process called interoception. Interoception refers to the brain's ability to sense the internal condition of the body. While the external senses provide information about the outside world, interoceptive signals inform the brain about what is happening inside. Sensors distributed throughout the body monitor variables such as heart rate, breathing, blood pressure, body temperature, and the chemical composition of the bloodstream. These signals travel through neural pathways to regions of the brain that monitor the body's internal state. A key region involved in interoception is the insular cortex. The insula integrates signals from across the body and contributes to the subjective feeling of bodily states. When your heart beats faster during excitement, when hunger begins to emerge, or when fatigue gradually settles in, the brain becomes aware of these internal changes through interoceptive processing. These signals do more than simply report the condition of the body. They also influence emotional states, decision-making, and attention. For example, the feeling of anxiety often involves a combination of cognitive interpretation and bodily signals such as increased heart rate and muscle tension. The brain interprets these internal cues as part of the emotional experience. Interoception therefore plays a crucial role in linking physical states with mental experience. Another intriguing example of brain-body interaction can be found in the relationship between the brain and the digestive system. This connection is often referred to as the gut-brain axis. The digestive system contains an extensive network of neurons known as the enteric nervous system. Sometimes informally called the second brain, this system contains tens of millions of neurons embedded within the walls of the digestive tract. These neurons regulate processes such as digestion, nutrient absorption, and intestinal movement. But they also communicate extensively with the central nervous system. Signals travel between the gut and the brain through several pathways, including the vagus nerve, hormonal signals, and immune system interactions. Neuroscientist John Cryan and his colleagues have studied how microorganisms living in the digestive system influence this communication. The human gut hosts trillions of microbes forming a complex ecosystem known as the gut microbiome. These microbes produce chemical compounds that can affect neural signaling and immune responses. Research suggests that changes in the gut microbiome may influence mood, stress responses, and even certain cognitive functions. Although scientists are still investigating these relationships, the findings highlight how deeply interconnected the brain and body truly are. Taken together, these discoveries challenge the idea that the mind exists solely within the brain. Instead, cognition appears to emerge from a distributed system, involving neural circuits, bodily processes, and environmental interactions. The brain interprets signals from the senses. The body provides internal feedback about physiological states. The environment presents opportunities and challenges that shape perception and action. Through the continuous exchange of information across these domains, the mind unfolds. Thoughts arise within neural networks, but those networks are constantly shaped by signals from the body and by interactions with the world. Every breath, every movement, every change in the environment subtly influences the patterns of activity within the brain. The mind, therefore, is not merely something contained inside the skull. It is the dynamic result of a living system in motion, a system in which brain, body, and world remain inseparably linked as experience continues to unfold. At any given moment, your senses are delivering an astonishing amount of information to your brain. Light reflecting from countless objects enters your eyes. Sounds from many directions reach your ears. The skin constantly reports pressure, temperature, and movement. Meanwhile, internal signals from the body continue to stream upward from organs, muscles, and the cardiovascular system. If the brain attempted to process every piece of this incoming data with equal priority, the result would quickly become overwhelming. The nervous system would drown in its own sensory input. Instead, the brain relies on a powerful mechanism that acts as a kind of filter. This mechanism is attention. Attention allows the brain to select certain pieces of information for deeper processing, while temporarily ignoring others. It helps determine which signals become the focus of awareness and which fade quietly into the background. Without attention, perception would be chaotic. Every stimulus would compete equally for processing, leaving the mind unable to focus on what matters most. Consider a familiar situation. You might be sitting in a busy cafe filled with conversations, clinking dishes, music playing quietly in the background, and footsteps moving across the floor. Despite this complexity, you're usually able to follow a conversation with the person sitting across from you. Your brain accomplishes this by directing attention toward the voice of your companion while reducing the influence of other sounds. This selective process is sometimes referred to as the cocktail party effect, and it illustrates how attention shapes perception. But how does the brain decide which signals deserve priority? One influential explanation comes from a theory known as biased competition. This idea was developed by neuroscientists Robert Desimone and John Duncan. Their theory proposes that objects and stimuli in the environment compete with one another for representation within neural networks. At any moment, multiple stimuli activate overlapping groups of neurons in the visual system and other sensory areas. These stimuli effectively compete for limited neural resources. Attention biases this competition. When attention is directed toward a particular stimulus, the neural signals representing that stimulus are strengthened. Competing signals representing less relevant stimuli become weaker. This process allows the brain to enhance information that is currently important while suppressing distractions. Imagine a spotlight moving across a stage in a dark theatre. Wherever the spotlight shines, the actors become clearly visible while the rest of the stage fades into shadow. Attention functions in a similar way within the brain. It enhances certain neural signals, bringing them into sharper focus while others remain dim. The ability to control this spotlight depends on specialized networks within the brain. Psychologist and neuroscientist Michael Posner conducted influential research identifying several distinct attentional systems. His work suggested that attention involves multiple networks working together rather than a single centralized mechanism. One network is responsible for alerting the brain to incoming stimuli. This alerting system helps maintain a general state of readiness, ensuring that important signals are detected quickly. Another network controls orienting attention. This system directs focus toward particular locations or objects in the environment. When you turn your eyes or shift your focus toward a sudden movement, this orienting system is at work. A third network supports executive control of attention. This system helps manage complex tasks that require concentration, such as solving problems or resisting distractions. These attentional networks involve interactions between several brain regions, including areas of the frontal cortex and parietal cortex. Together they help coordinate where attention is directed and how strongly certain signals are amplified. At the level of individual neurons, attention appears to change the strength of neural responses. When a stimulus becomes the focus of attention, neurons that represent that stimulus tend to fire more strongly. Their signals become amplified relative to competing inputs. This amplification increases the likelihood that information about the attended stimulus will be transmitted to other parts of the brain. In contrast, signals representing unattended stimuli may remain weak and may never reach higher processing stages. Because of this selective amplification, attention strongly influences what enters conscious awareness. Many sensory signals are processed to some degree within the brain without ever becoming part of conscious experience. For example, visual information about objects outside the center of your gaze may activate neurons in the visual cortex, yet you remain unaware of these details unless attention shifts toward them. Psychologists have demonstrated this phenomenon in striking experiments involving inattentional blindness. In one well-known demonstration, participants watch a video in which several people pass a basketball back and forth. The task is to count the number of passes made by one team. During the video, a person wearing a large gorilla costume walks slowly through the scene, pauses briefly, and then exits. Surprisingly, many viewers completely fail to notice the gorilla. Their attention is so strongly focused on counting the basketball passes that the unexpected figure never enters conscious awareness. The visual system detects the image, but without attention, it fails to become part of the perceived scene. This reveals something remarkable about the relationship between attention and consciousness. The brain may process far more information than we consciously experience. Attention acts as a gateway that determines which signals rise into awareness. In this sense, consciousness does not simply reflect everything the brain perceives. It reflects the information that attention selects and amplifies. Through this mechanism, attention allows the brain to operate efficiently in a complex world. Rather than attempting to process every signal with equal intensity, the nervous system concentrates its resources on the most relevant information. This selective focus allows us to read a sentence despite background noise, follow a conversation in a crowded room, or notice the sudden movement of a passing car. Attention therefore serves as one of the mind's most powerful organizing tools. It shapes perception, it guides thought, and it quietly determines which fragments of the sensory world become the experiences that fill our conscious awareness. Most of us move through life with a quiet assumption that feels almost impossible to question. The assumption is that there is a stable self inside the mind, a continuous individual who experiences the world, remembers the past, plans for the future, and remains fundamentally the same person from one moment to the next. Even as our bodies change and our thoughts shift, the feeling of being a single enduring self seems remarkably solid. Yet when neuroscientists and philosophers begin examining this sense of self more closely, an intriguing possibility emerges. The self may not be a fixed entity at all. Instead, it may be something the brain continuously constructs. One of the philosophers who has explored this idea in depth is Thomas Metzinger. Metzinger proposed what he calls the self-model theory of subjectivity. According to this theory, the experience of being a self arises from a model that the brain generates to represent the organism as a unified agent interacting with the world. In this view, the brain constructs a detailed internal model that includes information about the body, the environment, and the organism's goals and intentions. This model becomes so transparent that we do not experience it as a model. Instead, we experience it directly as ourselves. Metzinger describes this transparency as a key feature of the self-model. When the brain constructs a model of the body and the mind, the process itself becomes invisible to awareness. What remains is the feeling that there simply is a self present inside the experience. It is similar to looking through a perfectly clean window. You see the landscape outside, not the glass itself. In the same way, the brain's self-model allows us to experience the world from a particular perspective, while remaining unaware of the underlying construction process. This idea becomes even more interesting when we consider that the self may not be a single unified phenomenon. Philosopher Sean Gallagher has proposed that the sense of self can be divided into at least two related components. The first is the minimal self. The minimal self refers to the immediate sense of being an embodied subject of experience. It is the feeling that perceptions are happening to you right now, in the present moment. When you move your hand and feel it move, when you see the world from a particular viewpoint, or when you sense your body occupying space, you're experiencing the minimal self. This aspect of selfhood is closely linked to bodily awareness and sensory processing. The second component is the narrative self. The narrative self involves the story we tell about who we are over time. It includes memories, beliefs, goals, personality traits, and the sense of continuity that connects past experiences with present identity. When someone says, "I am a teacher," or "I grew up near the ocean," they are expressing elements of the narrative self. It is the autobiographical storyline that gives shape to personal identity. These two forms of selfhood operate together but rely on somewhat different neural processes. The minimal self arises from immediate sensory and bodily signals, while the narrative self depends heavily on memory and language. One of the most striking demonstrations of how flexible the sense of self can be comes from a series of experiments known as body ownership illusions. Perhaps the most famous example is called the rubber hand illusion. In this experiment, a participant sits at a table with one of their real hands hidden from view. In front of them lies a realistic-looking rubber hand positioned in a natural orientation. An experimenter then begins gently stroking both the hidden real hand and the visible rubber hand with a small brush. The strokes are synchronized so that both hands appear to be touched at the same moment. After a short period of time, many participants begin to feel as though the rubber hand actually belongs to them. Their brain integrates the visual information of the brush, touching the rubber hand with the tactile sensation from the hidden hand. Because these signals occur simultaneously, the brain infers that the rubber hand must be part of the body. If the rubber hand is suddenly threatened, for example, by bringing a small hammer close to it, participants often display physiological reactions such as increased heart rate or a reflexive withdrawal. Their brain has temporarily incorporated the rubber hand into its body representation. Experiments like this reveal that the sense of bodily selfhood is not fixed. Instead, it emerges from the brain's integration of sensory signals. The brain constructs a model of the body based on vision, touch, and proprioception, the sense of where body parts are located in space. When these signals align in certain ways, the model can shift. The feeling of owning a body is therefore not simply given. It is constructed moment by neural processes. Beyond bodily awareness, another network of brain regions appears to play a role in maintaining the narrative sense of self. This network is known as the default mode network. The default mode network was first described by neuroscientist Marcus Rachel and his colleagues while studying patterns of brain activity using functional brain imaging. They noticed that certain regions of the brain became highly active when participants were not focused on external tasks. Instead of shutting down during rest, these regions showed increased activity when people engaged in internal thought. The default mode network includes areas such as the medial prefrontal cortex, the posterior cingulate cortex, and parts of the parietal lobe. These regions become active during activities like recalling personal memories, imagining future events, reflecting on one's own personality, or thinking about other people. In other words, this network appears closely tied to the construction of the narrative self. When the mind wanders and begins reflecting on past experiences or future plans, the default mode network becomes particularly active. It helps integrate memories, emotions, and social understanding into the ongoing story we tell about who we are. Taken together, these findings suggest that the self may not be a single structure, located somewhere inside the brain. Instead, it appears to be the result of multiple interacting processes. Sensory systems construct a representation of the body. Memory systems maintain continuity with the past. Predictive processes anticipate future possibilities. Attention organizes what enters awareness. Through the interaction of these systems, the brain generates a coherent perspective that feels like a stable self. Yet beneath that stability lies constant change. Neural networks are continuously updating their models of the body and the world. New experiences reshape memory. Goals and beliefs evolve over time. The story of who we are is revised with every new chapter of life. In this sense, the self may be less like a fixed object and more like an ongoing simulation, a dynamic model maintained by the brain to organize perception, guide action, and maintain coherence across time. The feeling of being a self remains remarkably convincing. But beneath that feeling lies a living process, a process that quietly reconstructs the experience of identity moment by moment as the brain continues its endless work of making sense of the world. At any given moment, your brain is performing an extraordinary amount of work. Signals are flowing through billions of neurons, chemical messengers are being released at trillions of synapses, and networks across the brain are coordinating their activity in intricate patterns. Yet only a very small portion of this activity ever reaches conscious awareness. Most of the brain's operations unfold quietly behind the scenes, beyond the narrow spotlight of attention. These hidden processes analyze sensory information, regulate bodily functions, shape decisions, and prepare actions long before we become aware of them. In this sense, the conscious mind may represent only a small window into a much larger system of unconscious activity. Psychologist Timothy Wilson has described this idea using a memorable phrase. He refers to the unconscious mind as the adaptive unconscious. Wilson's research suggests that many judgments, preferences, and behavioral tendencies are influenced by mental processes that operate outside conscious awareness. The brain constantly evaluates information, detects patterns, and forms impressions without requiring deliberate thought. For example, within fractions of a second after meeting someone, people often develop subtle impressions about trustworthiness or friendliness. These impressions arise from rapid unconscious analysis of facial expressions, tone of voice, posture, and countless other cues. Although we may later try to explain these impressions using conscious reasoning, the initial evaluation often occurs automatically and unconsciously. The unconscious mind also helps manage complex skills that would be difficult to perform if we had to consciously control every step. Consider riding a bicycle. When someone first learns to ride, they must consciously think about balance, steering, and pedaling. But after enough practice, the skill becomes automatic. Neural circuits in the brain and spinal cord handle the coordination without requiring constant conscious attention. The same principle applies to many aspects of everyday life. Walking through a familiar environment, understanding the grammar of a language, recognizing faces or interpreting emotional expressions. These tasks rely on sophisticated neural processing that rarely enters awareness. But perhaps one of the most intriguing demonstrations of unconscious processing comes from experiments investigating how decisions are made. In the 1980s, neuroscientist Benjamin Libet conducted a series of experiments that raised fascinating questions about the relationship between conscious intention and brain activity. Libet asked participants to perform a simple task. They were instructed to move a finger whenever they felt the spontaneous urge to do so, while also noting the moment they first became aware of the intention to act. At the same time, researchers recorded electrical activity in the participants' brains using electroencephalography. The results revealed something surprising. A specific pattern of neural activity known as the readiness potential appeared in the brain several hundred milliseconds before participants reported the conscious decision to move their finger. In other words, the brain seemed to begin preparing the action before the person became consciously aware of deciding to act. These findings sparked intense debate among scientists and philosophers. Some researchers interpreted the results as evidence that unconscious brain processes initiate actions before conscious awareness enters the picture. Others argued that consciousness might still play a role in approving or vetoing actions after the initial neural preparation. Regardless of the interpretation, Libet's experiments highlighted an important point. The brain's decision-making processes may begin at levels that operate outside conscious awareness. Conscious thought may not be the sole driver of behavior, but rather part of a broader system in which unconscious and conscious processes interact. Understanding how conscious awareness fits into this larger system has become a major focus of modern neuroscience. One influential framework addressing this question is called Global Neuronal Workspace Theory. This theory was originally proposed by cognitive scientist Bernard Baars and later developed further by neuroscientist Stanislas Dehaene and his colleagues. According to Global Neuronal Workspace Theory, the brain contains many specialized processing systems that operate largely independently. Different networks handle visual analysis, auditory processing, memory retrieval, emotional evaluation, and motor control. Most of these systems process information locally within their own circuits, without entering conscious awareness. However, when certain information becomes particularly relevant or important, it can gain access to a global workspace. The global workspace acts like a broadcasting system within the brain. When information enters this workspace, it becomes widely available to multiple brain regions at the same time. This widespread broadcasting allows different neural systems to coordinate their activity using the same shared information. Conscious awareness may correspond to the moment when information is broadcast across this global network. To illustrate the idea, imagine a large organization with many specialized departments working on different tasks. Most communication occurs within individual departments, but occasionally a message is sent through the central communication system so that everyone receives the same information simultaneously. When this happens, the information becomes globally accessible. Global neuronal workspace theory suggests that conscious perception operates in a similar way. Information that wins the competition for attention becomes amplified and broadcast across the brain's networks. Once broadcast, that information can influence memory, decision-making, planning, and verbal report. This framework helps explain why we can only focus on a limited number of things at once. The brain may process many signals unconsciously in parallel, but the global workspace can broadcast only a small amount of information at any given moment. The result is a conscious experience that feels focused and coherent, even though it arises from a vast landscape of unconscious activity. Taken together, these discoveries paint a remarkable picture of the mind. Beneath the surface of awareness lies an enormous network of neural processes continuously evaluating the world, guiding behavior, and maintaining the body's internal balance. Unconscious systems detect patterns, prepare actions, and integrate information long before we become aware of the results. Consciousness then appears to function as a kind of spotlight and broadcasting platform, bringing selected information into a shared workspace where it can influence many parts of the brain at once. The mind we experience each day may therefore represent only the visible portion of a much larger system. Behind the scenes, the unconscious brain is always at work, quietly shaping the flow of perception, thought, and action that becomes the story of conscious experience. For much of the 20th century, many scientists believed the structure of the adult brain was largely fixed. According to this view, the brain developed during childhood, established its network of connections, and then remained relatively stable throughout adulthood. Learning might adjust how existing circuits were used, but the underlying architecture was assumed to stay mostly unchanged. Over the past several decades, that assumption has been overturned. Modern neuroscience has revealed that the brain is remarkably adaptable. Its connections can reorganize, strengthen, weaken, and sometimes even grow in response to experience. This ability is known as neuroplasticity. Neuroplasticity refers to the brain's capacity to change its structure and function based on activity. Every time you learn a new skill, form a memory, or practice a task, the patterns of connectivity between neurons are subtly reshaped. In this way, experience leaves a physical imprint on the nervous system. The concept of neuroplasticity has roots that stretch back to the mid-20th century, when psychologist Donald Hebb proposed a principle that would later become one of the most influential ideas in neuroscience. Hebb suggested that when two neurons repeatedly become active at the same time, the connection between them grows stronger. This principle is often summarized in a phrase that has become famous in neuroscience: cells that fire together wire together. This idea describes a process known as Hebbian learning. When neurons consistently participate in the same pattern of activity, the synapses connecting them increase in strength. As those connections strengthen, the neurons become more likely to activate together in the future. Over time, repeated experiences can therefore shape the structure of neural circuits. The principle can be expressed in a simple conceptual form. The strength of a synaptic connection increases when the activity of the sending neuron repeatedly contributes to the firing of the receiving neuron. Although the real biology is more complex than this simplified statement, the general idea captures an essential feature of learning in the brain. Neural circuits are not static. They evolve based on patterns of activity. One of the scientists who demonstrated the power of plasticity through experimental research is neuroscientist Michael Merzenich. Merznick studied how sensory areas of the brain reorganize themselves in response to experience. In one series of experiments, he and his colleagues mapped the regions of the cortex that respond to touch on the fingers. They discovered that when animals repeatedly use certain fingers for specific tasks, the corresponding regions of the sensory cortex expanded. More neurons became dedicated to processing input from those frequently used fingers. In contrast, areas representing less active fingers could shrink or shift over time. These findings showed that the brain's sensory maps are not fixed. They adjust dynamically as patterns of activity change. Merznick's work also contributed to the development of therapies designed to help patients recover from brain injuries or adapt to sensory loss. By designing training exercises that encourage specific neural circuits to activate repeatedly, it is possible to promote beneficial reorganization within the brain. Plasticity therefore offers hope for rehabilitation after conditions such as stroke, traumatic brain injury, or sensory impairment. But plasticity presents an interesting challenge. If neural circuits are constantly changing in response to experience, how does the brain maintain stability? After all, essential functions such as breathing, balance, and motor coordination must remain reliable even as the brain adapts. This question has been explored by neuroscientist Eve Marder. Marder studies neural circuits in relatively simple nervous systems, particularly those controlling rhythmic behaviors such as the movements of crustacean digestive systems. Although these systems are far less complex than the human brain, they reveal important principles about how neural circuits maintain stability. Her research has shown that individual neurons can vary widely in their internal properties, yet the overall circuit can still produce stable behavior. In other words, the components of the system may change, but the collective function remains consistent. This phenomenon is known as degeneracy in biological systems. Multiple different configurations of neural properties can produce the same overall output. Marder's work suggests that the brain balances plasticity with mechanisms that stabilize network function. While connections adjust through experience, regulatory processes ensure that circuits continue to operate within functional limits. The result is a system that is both adaptable and stable. It can learn from new experiences while preserving essential abilities. Plasticity operates at multiple levels within the brain. At the level of synapses, connections between neurons can strengthen or weaken depending on patterns of activity. This process involves changes in neurotransmitter release, receptor sensitivity, and the structure of synaptic contacts. At a larger scale, neurons can grow new dendritic branches or form new synaptic connections with neighboring cells. Even entire neural networks can reorganize when sensory input changes or when new skills are learned. Consider the example of learning to play a musical instrument. During the early stages of practice, the task may feel awkward and demanding. The brain must coordinate complex movements of the hands, interpret auditory feedback, and integrate visual information from musical notation. With repeated practice, neural circuits involved in these tasks gradually strengthen and reorganize. Connections become more efficient, movements become smoother, and the performer requires less conscious effort to produce the desired sounds. Brain imaging studies have shown that experienced musicians often display measurable differences in the structure and activity of brain regions involved in auditory processing and motor control. These differences reflect years of plastic changes shaped by training. Similar processes occur whenever we acquire new knowledge or skills, learning a language, navigating a new city, practicing a sport, even forming a new habit. Each experience contributes to subtle modifications in the brain's network of connections. In this sense, every moment of life participates in shaping the physical structure of the brain. The patterns of activity flowing through neural circuits leave traces that influence how those circuits function in the future. Experience becomes architecture, memory becomes wiring, and the mind itself gradually evolves as the brain continues to reorganize in response to the world. The remarkable capacity for plasticity ensures that the brain remains a living, adaptable system, not a fixed machine, but a constantly changing network whose structure reflects the story of every experience it has ever encountered. Each night, as the body settles into sleep, the brain begins a journey into a very different mode of activity. The outside world fades from awareness, muscles relax, breathing slows, and consciousness drifts into a state that feels quieter and more distant. Yet the brain itself does not simply shut down. In many ways, it becomes surprisingly active. During sleep, the brain cycles through several stages that repeat throughout the night. These stages include periods of lighter sleep, deeper slow-wave sleep, and a distinctive phase known as rapid eye movement sleep, often abbreviated as REM sleep. REM sleep is the stage most strongly associated with vivid dreaming. The name rapid eye movement comes from a curious observation. When researchers began studying sleep using electroencephalography in the mid-20th century, they noticed that sleepers sometimes entered a phase where their eyes moved rapidly beneath closed eyelids. At the same time, brain activity recorded during this phase looked strikingly similar to patterns seen during wakefulness. Electrical signals across the cortex became fast and irregular, suggesting a high level of neural activity. Meanwhile, the body itself remained largely immobile. Muscles were temporarily inhibited by brainstem mechanisms that prevent the sleeper from physically acting out their dreams. In this unusual state, the brain generates elaborate internal experiences. Dreams can include complex visual scenes, conversations, emotions, and narratives that unfold with remarkable realism. People may find themselves walking through familiar cities, flying through impossible landscapes, or encountering individuals who have not been seen in years. Despite the vividness of these experiences, they arise entirely within the brain. The external world is largely disconnected during REM sleep, yet the mind continues to generate perceptions that feel almost as real as waking life. This remarkable ability highlights something fundamental about the brain. It does not require direct sensory input to produce rich experiences. The neural machinery that constructs perception can operate internally, generating entire worlds from memory, imagination, and spontaneous activity. Understanding why this happens has fascinated scientists for decades. One influential explanation emerged in the 1970s through the work of neuroscientists Alan Hobson and Robert McCarley. They proposed what became known as the activation synthesis theory of dreaming. According to this theory, dreaming begins with spontaneous activity originating in the brainstem during REM sleep. These signals activate various regions of the cortex responsible for perception, memory, and emotion. The cortex then attempts to interpret these internally generated signals, weaving them into a narrative that becomes the dream. In this model, the brain is essentially trying to make sense of its own activity. Random neural signals are synthesized into meaningful experiences because the brain is constantly searching for patterns and explanations. The result is the dream story that unfolds during sleep. Although this theory captures an important aspect of dreaming, more recent research suggests that dreams may serve additional functions beyond simply interpreting spontaneous brain activity. Neuroscientist Mark Solms has contributed significantly to modern understanding of dreaming through careful studies of patients with brain injuries affecting different regions involved in sleep and emotion. Solms discovered that damage to certain areas of the brainstem did not eliminate dreaming, as earlier theories had predicted. Instead, dreaming appeared closely linked to structures involved in motivation and emotional processing. In particular, networks associated with emotional drives and memory appear to play important roles in shaping dream experiences. Solms proposed that dreams may reflect the brain's attempt to process emotional concerns and unresolved experiences. The mind may use dreaming as a way to explore emotional situations, rehearse responses, or integrate memories. Although researchers continue to debate the precise function of dreaming, most agree that dreams demonstrate an extraordinary feature of the brain. It can simulate reality. During dreaming, the same neural systems that produce waking perception become active in new combinations. Visual areas generate imagery. Memory systems contribute fragments of past experiences. Emotional circuits color the events, with feelings ranging from joy to fear. Together, these systems create immersive internal worlds. Interestingly, many of the brain regions responsible for logical reasoning and self-monitoring become less active during REM sleep. This may explain why dreams often contain strange events that would seem impossible in waking life. A person may suddenly fly through the air or encounter people from different periods of their life, appearing in the same scene. The dreaming brain accepts these events without question, because the systems responsible for critical evaluation are less engaged. Yet despite their unusual logic, dreams often feel deeply meaningful while they occur. The same neural circuits that support imagination during waking life appear to contribute to dream generation. Both processes involve the brain's ability to recombine stored memories into new scenarios. When you imagine a future event, the brain draws upon fragments of past experiences and rearranges them into a possible situation. Dreams perform a similar process, but without the constraints imposed by the external environment. In this sense, dreaming reveals something profound about the nature of the mind. The brain is not merely a device that records the world, it is also a generator of experiences. It can construct vivid scenes, emotional narratives, and sensory worlds entirely from internal activity. Every night as sleep unfolds, the brain demonstrates this creative capacity. Within the quiet darkness of the sleeping mind, entire landscapes appear, characters speak, events unfold, and the brain continues its remarkable work of generating the experience we call reality, even when the outside world is temporarily left behind. After exploring neurons, networks, perception, memory, emotion, and the many processes shaping the mind, one question still lingers quietly beneath everything else. Why does any of this activity produce experience at all? Neuroscience has made enormous progress in understanding how the brain works. Scientists can record the firing of neurons, trace communication between brain regions, and observe how patterns of activity change during perception, learning, or sleep. Researchers can identify what are called neural correlates of consciousness. These are patterns of brain activity that reliably accompany conscious experiences. For example, certain regions of the visual cortex become active when a person sees a face. Particular networks become active when someone recalls a memory or focuses attention on a task. But identifying these correlations does not fully explain the deeper mystery. The firing of neurons can be measured. Electrical signals and chemical exchanges can be observed. Yet none of these measurements directly reveal why the activity should feel like anything from the inside. The sensation of seeing the color blue, the feeling of warmth from sunlight on your skin, the quiet awareness of your own thoughts. These subjective qualities are often called qualia, the raw textures of conscious experience. Understanding how physical processes give rise to these experiences remains one of the greatest unsolved questions in science. Several theories attempt to address this puzzle. One of the most ambitious proposals comes from neuroscientist Giulio Tononi. Tononi developed a framework known as integrated information theory. Integrated information theory begins with a simple observation. Conscious experiences appear to be both unified and richly structured. When you perceive a scene, you do not experience separate fragments of color, shape, and motion floating independently. Instead, these features are integrated into a single coherent experience. At the same time, that experience contains a vast amount of information. A single glance at a room reveals countless details about objects, colors, spatial relationships, and movement. Tononi proposed that consciousness may arise in systems that both integrate information and generate a large number of possible states. To capture this idea, integrated information theory introduces a quantity known as phi, spelled phi. Phi represents the degree to which a system integrates information across its components. In principle, a system with higher phi would possess a greater capacity for unified experience. Tononi suggests that consciousness corresponds to the amount of integrated information generated by a system's internal structure. In this framework, the brain supports consciousness because its network of neurons is highly interconnected. Signals flowing through these networks combine information from many sources into unified patterns of activity. Although the mathematics behind integrated information theory is complex, the central idea is conceptually simple. Consciousness may arise when information becomes deeply integrated within a system. Another influential explanation takes a somewhat different approach. Global workspace theory focuses less on the intrinsic structure of information and more on how information becomes widely available within the brain. Originally proposed by cognitive scientist Bernard Baers and later developed by neuroscientist Stanislas Dehaene, global workspace theory suggests that many specialized neural processes operate simultaneously within the brain. Most of these processes remain unconscious. However, when certain information becomes especially relevant or strongly activated, it gains access to a global workspace. Once inside this workspace, the information is broadcast across multiple brain regions. This broadcast allows different systems involved in perception, memory, language, and decision-making to access the same information. In this theory, consciousness corresponds to the moment when information becomes globally available within the brain's networks. A perception becomes conscious when it is amplified and shared widely across neural circuits. While integrated information theory focuses on the degree of information integration, global workspace theory emphasizes the widespread distribution of information. Both frameworks attempt to explain the conditions under which conscious awareness emerges from neural activity. Yet even these sophisticated theories leave an important philosophical question unresolved. Why should any physical process generate subjective experience in the first place? Some philosophers have suggested that our current scientific framework may be missing an essential piece of the puzzle. One such thinker is philosopher Philip Goff, who has explored a modern version of an idea known as panpsychism. Panpsychism proposes that consciousness, or at least very simple forms of experience, may be a fundamental feature of the universe, rather than something that emerges only in complex brains. According to this view, basic elements of matter might possess extremely simple experiential properties. These elementary forms of experience would not resemble human consciousness, but they might represent primitive building blocks from which more complex forms of awareness could arise. In such a framework, the human brain would not create consciousness from nothing. Instead, it would organize simpler experiential components into the unified consciousness we experience. Although panpsychism remains controversial, it illustrates how scientists and philosophers are exploring unconventional possibilities in order to address the mystery of consciousness. Another philosophical perspective related to this idea is known as neutral monism. Neutral monism was discussed by philosopher Bertrand Russell in the early 20th century. Russell proposed that the fundamental substance of reality might be neither purely mental nor purely physical. Instead, the basic building blocks of the universe might possess properties that give rise to both mental and physical phenomena under different conditions. In this framework, matter and mind would not be completely separate categories. They would be different aspects of a deeper underlying reality. While these philosophical ideas remain speculative, they highlight the limits of our current understanding. Neuroscience has made remarkable progress in mapping the brain's activity and identifying the neural processes associated with perception, memory, and decision-making. Scientists can observe how patterns of activity shift as a person becomes conscious of a stimulus or loses awareness during sleep or anesthesia. But the deeper question remains open. Why does any of this activity feel like something from the inside? Why does a particular arrangement of neurons and electrical signals produce the experience of a color, a sound, or a thought? At present, no scientific theory has fully answered this question. The brain clearly plays a central role in generating conscious experience. Damage to certain regions can disrupt awareness, while stimulation of other regions can evoke vivid sensations. Yet the precise relationship between physical processes and subjective experience remains mysterious. This mystery does not diminish the progress that neuroscience has made. Instead, it reminds us that some of the most profound questions about the mind are still waiting to be answered. Inside the 3 pounds of living tissue within the skull, billions of neurons exchange signals every moment. From this activity emerges perception, thought, emotion, and the quiet sense of being aware. Exactly how that transformation occurs remains one of the greatest scientific frontiers, and the search for its explanation continues to unfold.